In this session, we're going to talk about biomolecules, which are also called organic molecules or organic compounds. Now, we've talked about atoms being the smallest, particle of an element or of an organism. Now, we're also still talking about small molecules, but slightly larger than atoms. So we're going to talk about biomolecules, which are small. Then we are going to proceed further. We're going to talk about larger biomolecules. So we start with small biomolecules like amino acids, fatty acids, nucleotides. Now you discover that these biomolecules have got what we call functional groups. That's why it's imperative for us to also talk about functional groups and what a functional group is. You know about a functional group as we proceed further. Then we're going to introduce the first part of the smallest biomolecules, which are our monosaccharides in this case, in terms of the carbohydrates. Now let's proceed. Now, what is an organic molecule or what is a biomolecule? And what is an inorganic molecule? Now, an organic molecule in this case, we say it's an element or it's a compound that contains a carbon atom or an element of carbon. Then those compounds or those elements that do not have carbon either in their structure or in their in their morphology, we say they are inorganic. So these ones, they don't have a carbon atom. Now, like the human body itself contains both organic and inorganic compounds. Now, how many electrons does a carbon have in its outermost shell? If we are going to draw, this is our nucleus. Okay, let me say this is our first shell. We'll not draw the nucleus. This is our second shell. Now, you know that carbon is number 6, so the first shell will only accommodate 2 electrons. Then the second shell has the ability to accommodate 4, in this case, the last 4 that remain. So from this structure, you can see that carbon has an ability, or it can form 4 covalent bonds with other elements, and carbon forms covalent bonds. And like we earlier said in our first session, covalent bonds occur usually between nonmetals, and there is an equal sharing of electrons. Now, most elements that carbon will bond to, we can give the common examples that we can give. We can say, nitrogen. Carbon can also bond to hydrogen. Carbon can bond to, a hydrogen. Carbon can also bond to a fellow carbon molecule. Now, all these small molecules that we're talking about, like amino acids, sugars, fatty acids, and nucleotides, all of them have a carbon backbone. But in addition to that carbon backbone, they have what we call functional groups. A functional group such what we call like a hydroxyl, a carboxyl, a phosphate group. Now let's talk about these functional groups. So these functional groups usually determine the characteristics of these small organic molecules. One feature of one organic molecule is that whether it is hydrophobic or hydrophilic, it depends on the functional group that it has. Let's start with the first one that we can talk about. Okay, what if we've got something like this? So this functional group, where you have what we call a carbonyl that is bonded to a hydrogen and another molecule, is called an aldehyde. Then we've got another molecule. Let's say we've got R. This R is bonded to carbon. Carbon is bonded to another molecule. Then there's a double bond between carbon and oxygen. This compound or this molecule will be called a ketone. This functional group is called a ketone. Then let's say you have another one where you have R in this case meaning any molecule that is connected to this. If you got a molecule that is attached to an OH, or what we call a hydroxide or a hydroxyl group, so this one is called an alcohol group or an alcohol functional group. Let's talk about other ones. If you've got carbon which is double bonded to oxygen, this one is called a carbonyl. Then if you have okay, where you have minus OH there, this one is called a hydroxide. You remember this when you're doing acids and bases, or you can also call it a hydroxyl group. Now there's also another one where you have phosphorus double bonded to oxygen. This side there's an OH. This side you have O and you have R. Then this side you've got another OH. Now this functional group is called a phosphate. Okay. Now you should note that all the functional groups that end in OH, or that have this OH functional group, they are hydrophilic. Hydrophilic to mean just hydro means just water. Philic just means to love. So they are hydrophilic, they are water loving. That's what we're talking about. Okay. Now let's just talk about some, because we're talking about monosaccharides, and monosaccharides are carbohydrates. So what are the general functions, or let me talk about some of the general characteristics of carbohydrates. Carbohydrates, even the name carbon, just coming from carbon, then hydrates meaning water. It means these ones are hydrates of carbon. And the general formula for carbohydrates, you have C, H2O, and the Y. Now the X and the Y, these ones are just variables. Now let's talk about these carbohydrates. You realize that carbohydrates are widely distributed in plants and animals like that, where they act as structural they act for a lot of structural molecules. They can be structural molecules. They can also perform some of other metabolic functions of the cell. Now the bulk of animal carbohydrates is usually derived from plants, and you know that plants usually make they make energy. Chlorophyll, in the presence of chlorophyll and sunlight. Okay, this formula is well known. This is a formula for what we call photosynthesis, the process by which plants make their own food. Now you realize that when we consume these plants, they contain a lot of these carbon molecules and atoms, and that carbon is now incorporated into our system. However, animals can also synthesize some of their carbohydrates from fats and proteins, but that's not the scope for this session. Now why are we talking about carbohydrates? Because of their importance. You realize, like, I'll pick one of the carbohydrates like glucose. Glucose is the most important carbohydrate in this case, because for us as humans, our brain needs carbohydrates. Like, it really needs carbohydrates or glucose for it to function properly. Now let's talk about some of the sugars. Now sugars can be classified as monosaccharides, disaccharides, and polysaccharides. Monosaccharides, you've got examples like glucose, you've got examples like fructose, you've got examples like galactose. These are monosaccharides. Then when you combine these monosaccharides, they'll give you what we call disaccharides. Di to just mean two. Then polysaccharide, these ones just means many of them, like that, many units. Now, if you combine glucose, a monosaccharide, and another monosaccharide glucose, you form what we call a maltose. Now, when you combine glucose and fructose, you form what we call a disaccharide called sucrose. Then when you combine glucose and galactose, you form, a disaccharide called lactose. Now, we'll discuss much of this when we start doing disaccharides. Now, let's talk about these, carbohydrates one by one. A monosaccharides, we'll just talk about them briefly. Now, monosaccharides, like we've said, these cannot be hydrolyzed into simpler, carbohydrates. For example, glucose. Even if you put glucose in water, it won't break, because it's the smallest, it's the simplest form of carbohydrate. Then you realize that these monosaccharides, they are made up of single units which are called monomers, like glucose monomers, it can be fructose monomers. Now, monosaccharides are used by cells as a direct source of energy. As you know, most of these carbohydrates, they are energy, giving foods, or energy giving, let me say, elements. Then disaccharides, these produce two molecules of monosaccharides when hydrolyzed. For example, the maltose that we got when you're combining glucose plus glucose, when we put this in water, it will hydrolyze to give two glucose molecules. Then we've got what we call an oligosaccharide. Now, oligosaccharides, they will yield what we call 2 to 10 monosaccharides. Those are oligosaccharides. An example that you can give under oligosaccharides is what we call maltotriose. So that's an example that you can give on oligosaccharide. Then let's talk about polysaccharides. Polysaccharides, these ones, they yield more than 10 molecules or monosaccharides units. So more than 10 monosaccharide units. When they're an example that you can give for a polysaccharide is starch. Now we're going to talk about all these ones one by one, but in this session, let's focus on monosaccharides. Now, monosaccharides contain, they will contain, carbohydrates. I would say they contain, especially like monosaccharides, they contain a carbon, they contain, a hydrogen and an oxygen. These usually occur in the ratio 1 to 2 to 1. So you have 1 carbon, 2 hydrogens, and that 1 oxygen. So hence the general formula for monosaccharide, or the empirical formula, the simplest whole number ratio will be CH2, CH2O. Then it depends with the number of carbons that it has, N to represent the number of carbons. So now you realize that monosaccharides will be classified based on the number of carbons. Now let's talk about this number of carbons. So we've got what we call a triose. Now a triose will have 3 carbon atoms. Then we've got what we call a tetrose. Now a tetrose will have 4 carbons. We've got what we call a pentose. A pentose will have 5 carbons. We've got what we call a hexose. Or a hexose will have 6 carbons, like that. Now, of these ones, you realize like all the names are ending in O's or OSE. OSE, OSE, OSE. Now, that is to mean that, monosaccharides, they end with the suffix or SE, or carbohydrates, they've got an OSE at the end. All monosaccharides have that OS E. Then all monosaccharides, glucose, like fructose, galactose, all of them, they are reducing sugars, meaning they are able to reduce copper to ions. Then this monosaccharide can exist as chains or rings, the simplest being the one that we just mentioned, the trios. Now let's talk about the trios, what a trios is. Now there are basically two trios that you need to know. We've got what we call a glycerolaldehyde and a dihydroxyacetone. Now a glycerolaldehyde, if you look at the structure, it's a trios. Yes, it has 3 carbons, 1, 2, 3. Now we're saying this is 1, this is 2, this is 3. Then from your organic chemistry, you can also name this compound in terms of organic chemistry. You realize that on carbon number 2 we have an OH functional group. On carbon number 3 also have another OH functional group. So the other name for a glycerolaldehyde would be 2, 3. Now because we've got 2 hydroxyl group, so we say dihydroxy or dihydroxyl propanone or propanol. Now propanol to me is an alcohol. You remember from your organic chemistry, propane, in terms of alkanes, propane had 3 carbons. Then if you had butane had 4, pentane had 5. So that's what we're talking about. Now this molecule has 3 carbons, so it will be like a propane. Now because it has OH groups, it will be a propane, and in this case we have a trios. Okay, let's just try to rub. So that's the other name for a glycerolaldehyde. Now before we move on to the dihydroxyacetone, I wanted to mention something. Now look at the structures of these two molecules. A dihydroxyacetone or a glycerolaldehyde, it has a carbonyl group at the end of the molecule. Then a dihydroxyacetone, it has a carbonyl group in the middle of the molecule. Now, like when we're talking about functional groups, what did we say when a molecule has a functional group at the end of the molecule? We said that this one is an aldehyde. Then so this one will be called the what? A ketone. So you notice the difference. The carbonyl, this one is at the end. The carbonyl, this one is in the middle of the molecule. Now a sugar with an aldehyde group or a carbonyl or a carboxyl group at the end of the molecule is called an aldose. So like this one, we say it's an aldose because it has an aldehyde group. So this one will be an aldose sugar. Then a glycerolaldehyde, because it has a ketone group, a sugar with a ketone group is called a ketose group. The carboxyl group is away from the end of the molecule, it's inside the molecule. So this one is called a ketose sugar. Then let's talk about something very, very important, what we call isomerism. Now, before I talk about isomerism, there's what is called an asymmetric carbon or a chiral carbon. If this is a carbon, this carbon is bonded to other molecules or elements. If the carbon is bonded to 4 different molecules, that one is called an asymmetric carbon. A carbon atom connected and surrounded by different formula units is called an asymmetric carbon or a chiral carbon. So let me say probably here is connected to OH. On the other side, let me say it's connected to H. Okay, this is just an example. Let me say NH3 Probably this side is also connected. Maybe let me say 3H or H. Now these groups are different. You've got a hydroxide there, you have a hydrogen, you've got a carbon, you've got like that. When these groups are different, it's called a chirocarbon. Now let's just rub a bit. Okay. So I thought of mentioning that, very, very important. So the opposite of a chirocarbon would be just a non-chiro carbo, or you call it a non-asymmetric carbon. Okay. Now let's talk about what we call isomerism. Now, isomerism, we're saying there are two types: a structural isomerism and stereo isomerism. Now, isomerism, just deriving the name an isomer. Now, what is an isomer? An isomer, these are molecules with the same atoms but arranged differently. So you find that the atoms are same. We're talking about the atoms will be the same, but you realize that the molecules themselves, they'll be arranged like differently. So they'll be differently arranged. So that one is called an isomer. Same atoms but arranged differently. Then we've got what we call structural isomers and stereo isomer. Then what is a structural isomer? So a structural isomer is an isomer that has the same number, that has the same type of atoms, but now those atoms are connected differently. I repeat, it has the same number of the number is the same, same number of atoms or same type, but now these ones are connected differently. So that is a structural isomer. Then we've got what we call a stereoisomer. Now stereoisomers, they have the same number of atoms as well. The same number of atoms, the same type of atoms. Then the atoms are even connected the same. On this one, the atoms are connected the same way, but the way the spatial configuration differs. The way they are configured really differs. So there's what we call the L and the D configuration. So these ones will be non-superimposable mirror images of each other, meaning you can put them together because they are mirror images of each other. Now, why does this happen? That's why I introduced the aspect of a chirocarbon. A chirocarbon, a carbon that is connected to 4 different, functional units. It's the one that is behind stereoisomerism. Now, how does this one happen? Okay. How does this one happen? Okay, let me just rub here. Okay. So we are saying you can have the L and the D configuration. Example, you can have L glyceroldehyde, D glyceroldehyde. Now, like for these ones on carbon number 2, we'll determine whether it's D or L, and some things like that. Now, this L and D will come in because of what we call, when you pass a beam of polarized light, it's passed through this maybe a solution of an optical or an isomer. You find that it will be rotated either to the right or to the left. When it's rotated to the left, it's called levorotatory. It will be called levorotatory conformation. Then when it's to the right, it will be called dextrorotatory. Okay, let me just try to write the spelling just for dextrorotatory. Okay, so this is to the right. This is the levorotatory is to to the left, hence just the L, just to help you remember. Now this is what we're talking about. In this case, we are using, a ribose, a 5 carbon sugar. So now you've got the D and the L. Now look at the D. All the hydroxides, they are on your right. All the hydroxides, they are on your right. Okay. Then for the L or the laboratory, they are on your left. So now these ones are called non-superimposable mirror images of it. It's like you put a mirror, but now the other part is reflected. So that's what these ones are just trying to show. So because of now, this happens because of what we call because of the asymmetric carbon, the carbon that is connected to 4 different groups. Yes. So for example, the carbon on number 2. The carbon on number 2 is connected to hydrogen, it's connected to a hydroxide, it's connected to this whole group, it's also connected to the to the carboxyl group there on top. So because it has because it has an asymmetric carbon or a chiral carbon, so what happens is that it tends to show isomerism, in this case stereoisomerism, where there are non-superimposable images of each other. Same with glucose here, the OH on the other side, like that. So that's what we're talking about. Now, how do you find the number of isomers of either an aldehyde or a ketone? Then just to remind you again, an aldehyde, you have a carbon, this is connected to an oxygen, there's a group here, there's also an H there. Then a ketone, we said there's a carbon here, there's a group this side, there's a group this side, then here there's a double bond with oxygen. Now we're saying an aldehyde, how do you find the number of isomers? So for an aldehyde, you say it's 2 to the power n minus 2. Then for a ketone, you say 2 to the power n minus 3. Okay, let's look at an example. Okay, an example that you can give is let's say this glucose. The glucose, let's just take one of these glucoses. Okay, this glucose. Okay, now you realize that this glucose structure, this is a straight structure. It has an aldehyde group at the end of the molecule. It has an aldehyde group at the end of the molecule. Then we are saying for you to find the number of isomers for this glucose molecule, you say 2n minus 2. N to represent the number of carbon atoms of that compound. So in this case you have 2n. N in this case, since it's glucose, how many carbons does it have? 6, right? You've got 1 here, you've got 2 here, 3, 4, 5, and 6 this one. So to be 6 minus 2. 2 to the power 6 minus 2, that is 4. 2 to the power 4, that is 2 times 2, 2 times 2, 4 times 4, that is 16. So glucose will have 16, isomers in this case, it being an aldehyde. That's what we're talking about. Okay. Then let's talk about what we call hexoses. Like we said, we are going to talk about trioses, hexoses, and pentoses. We've talked about trioses, glyceraldehyde, and dihydroxyacetone. Now let's talk about hexoses. Hexoses, these are monosaccharides with 6 carbons. Examples like we gave glucose, which had 6 carbon atoms. Then all of them have the simple formula of, all hexoses have the simple formula of C6, C6H12 or 6 or 6, like that. Then these hexoses, can be represented in 3 ways. It can either be a straight chain like the one we saw for glucose, a ring form, and a chain form. But mostly in this course you are going to talk about the straight chain and the ring form. Rarely do you talk about the chair form. Now glucose forms a ring, or glucose forms a ring called a pyranose. Glucose forms a ring called a pyranose. Now a pyranose, in this case, it is the stable form that glucose will be in, and it will have 6 sides like that. Then on the other hand, there's what we call a furanose ring. Now a furanose, this ring will be very stable for a compound, example, a molecule like fructose. This will be very stable, and this is a pentagon structure. It has 4 sides, so it will be very stable for fructose. Now why is this so? A pyranose will be very stable for like glucose will be very stable, because glucose, like we saw earlier, the carboxyl group is at the end of the chain, so glucose is an aldose. So we can also conclude that pyranose structure, they are stable. They're usually stable for aldoses, or a glucose or a sugar with an aldose group. Then for furanose, these ones they're stable in ketoses or a ketose. In this case, an example is fructose. So those are the things. Now let's proceed. Now there's what is called an alpha and a beta, alpha and beta. Now if the hydroxide group on carbon number 1 is below the ring, this will form what we call an alpha glucose. For example, this is the ring structure of glucose. Now this is carbon number 1, this is carbon number 2, this is carbon number 3, carbon number 4, carbon number 5, and carbon number 6. Now this ring has formed because the carbon that was on position number, the hydroxide that was on position number 5 reacted to break that bond on carboxyl number on carbon number 1. So that's how this ring forms. Now we are saying if the hydroxide, this hydroxide on carbon number 1, this one, it is below, then this one is called an alpha. In this case, is to be an alpha glucose. Then on the other hand, if the hydroxide on carbon number 1 is above the ring, like in this case, this one will be called a beta glucose. That's what we're trying to talk about on this diagram. Now how does the ring form? Let's just take, for example, now this is a pentose, a ribose sugar. Okay, it has 5 carbons. So now the carbon on number 4, it will go and react on the carbon, the hydroxide on position number 4 goes to react with the double bond there, the carbon that is has a double bond on carbon number 1. That's how this ring forms. Now it can either be a beta or an alpha. A beta, we've said, when the hydroxide is on top like that, on carbon number 1 here. Then an alpha when the hydroxide on carbon number 1 is down or below. That's what we're talking about in this case. Okay. Now let's talk about what we call epimers. Now, epimers, these differ in configuration of hydroxide and hydrogen on carbon atoms 2 and 4 of glucose. So they differ with glucose on carbon number 2 and carbon number 4 of glucose. So for example, mannose is an epimer of glucose on carbon number 2. Galactose is an epimer of glucose on carbon number 4. So let's look at some of the the diagram. This is our glucose here. This is our glucose here. Now let's compare it with mannose. Now mannose on position number 2. This is 1. This is number 2. On position number 2, you've got an OH inside the ring. But when you look at the glucose molecule on position number 2, the OH is outside the ring. Now everything else is the same apart on position number 2. So we say, they differ in configuration at position number 2, hence they are called the epimers. Then on the other hand, we've got galactose here. If you look at, this is position number 1, position number 2, position number 3, and position number 4. Now on position number 4, we've got a hydroxide outside the ring in galactose. When you go to glucose on position number 4 here, you find that the hydroxide is below the ring. Now it's below the ring. That's where they differ on position number 4. That's why we said galactose is an epimer of glucose on position number 4, and mannose is an epimer of glucose on position number 2. Now you must remember these ones because I've seen a lot of questions come on you differentiating the two. Now we've talked about trioses, we've talked about hexoses. Now let's talk about pentoses. Now what is a pentose? What is a pentose sugar? A pentose sugar is a monosaccharide with 5 carbon atoms. 5 carbon atoms. And examples, the two key examples that we can talk about, there's a ribose and deoxyribose. Now both of these are stable in the furanose ring because they are 5, because they've got 5 carbons, and we said 5 carbons usually goes with a pentagon structure. That's the stable form that they'll accommodate like that. So even in this case, that's what we're talking about. Now deoxy and ribose. Now these ones, they you find that, for example, this one is deoxy, this one is a ribose. Now where do they differ? On position number 2, you are able to see that for the ribose, position number 2, the oxygen is there, but on deoxy the oxygen is not there, only have hydrogen molecules, but everything around is the same. So deoxy, an example like deoxygenated blood that doesn't have oxygen. So even in this case, the oxygen on that position is not there. Now you realize that these ribose sugars, they help in forming what we call RNA, ribonucleic acid. Then your DNA is formed from deoxyribose or deoxyribonucleic acid. So DNA is just a component of almost all cells. All cells have DNA, and these are very important molecules. That's why we need to first talk about the components that make up these molecules as we proceed. So this is another diagram showing a pentose. In this case, this is the straight chain structure of a ribose. Then this is the ring form structure of a ribose. On position number 2, for a ribose, you have an OH, or there's an oxygen there, and we are saying ribose, they help in forming of RNA, as we'll be talking in other sessions. So even in the ring structure on position number 2, the OH is still there. But now for the deoxyribose on position number 2, they only have hydrogens because there's no oxygen in a deoxy. So this one helps in the formation of DNA, like we said. Then here on position number 2, again, only have hydrogens, this hydrogen and the other hydrogen. So this is what we can talk about today. Thank you. We'll see you in the next session where we're going to be talking about disaccharides.